Welcome to the University of Manitoba. My name is Dr. Peter Nickerson. I'm the Vice Dean Research for the Rady Faculty of Health Sciences. And before we get going, I just want to acknowledge that the U of M is located on the original lands of the Anishinaabe, Cree, Ojibwe Cree, Dakota, and Dene people, and on the homeland of the Métis Nation. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. Well, it's great to see people in person. I think it's the first meeting I've been here in 2.5 years, and I've been talking as the chair of the AFMC Vice Deans of Research. I've been meeting with all your grad deans and your Vice Deans through Zoom, as many of you have lived on Zoom. So it's great to actually have people here exchanging ideas and communicating their science to one another. And it's a pleasure to welcome you to the University of Manitoba on behalf of Dean Postal, who couldn't be here today, and to the 2022 Gairdner Symposium program. I've looked at the agenda. It's a fantastic program, unbelievable speakers on the cutting edge of science through all the pillars of CIHR. I think it's going to be a wonderful day. And I hope that you've been enjoying yourselves in Winnipeg and having a good time with the poster competitions. And I'm sure you're going to find out later in the day who all the winners are. Ed, thanks again for putting on such a great program for the country. And President Rosant, thanks so much for coming and with the Gairdner laureates to put on the program today. So again, have a fantastic day, everyone, and thanks for putting this on.
My welcome to everybody as well. I'm Jeff Hicks, and I'll be the MC for the day. I love to welcome you to the 35th Canadian Student Health Research Forum and the 6th Gairdner Foundation Symposium. You know, often some of the best discoveries and best science starts with a seemingly impossibly simple question and profound experiments to nail it after that. Today is not that. Today we're going to hear about great science that takes a look at significantly complex complexities underlying early development, health, and disease. So this morning, Dr. Lupien is going to describe how genetic drivers can guide precision medicine and cancer. Dr. Jocelyn will explore how mice make memories and how defects in that process can help explain autism and other neurodegenerative diseases. Dr. Rosant will explore early human development and really get into discussions of some of the complexities around the ethical framework of that research and new opportunities, new models that may get around those restrictions. And then this afternoon, Dr. Bada will really scale up this perspective to one of global health and community approaches to child and adolescent health. And then finally, Dr. Blanchard will chart new courses for science and global public health. So in addition to hearing research excellence and from some of the world's most inspired health researchers, we'll also hear a little bit about the journey from where they started in your seats today to where they stand before you today. So that will be another exciting component of this Gairdner Foundation Symposium. As Adam mentioned, this is the 6th Gairdner Symposium, and it's with great pleasure I invite Dr. Rosant up to give welcome words from the foundation itself. Janet.
Thanks, Jeff. It is a great pleasure to be here again on behalf of the Gairdner Foundation. I am the president and scientific director, as well as one of your lecturers today. It is 6 times we've been here, and it's been a wonderful opportunity. Ed invited Gairdner Foundation to become part of this event, bring Gairdner laureates, committee members, people associated with the Gairdner Foundation, just to really present some of the frontline research that goes on around the world. So for those of you who don't know what the Gairdner Foundation is, we are a foundation based in Toronto, but international in scope. And we give, our basic business is giving out awards, the Canada Gairdner Awards for biomedical research that's had impact on human health. The Canada Gairdner International Awards have been given out since the 1950s. The foundation was founded by Jim Gairdner, who was a Toronto philanthropist, who thought that scientists who were doing research didn't get enough recognition, and that it was very important to celebrate excellence wherever it occurs around the world that impacts human health. And that mission continues all the years since. We've given 402 prizes. 97 have gone on to get the Nobel Prize. And I joined the foundation in 2016, and every year since then we've had at least one and sometimes two Nobel Prize winners who are previous Gairdner awardees. So you've had at least one of those at your symposia. Bill Kaelin was here a few years ago before he got the Nobel Prize because he'd already received a Gairdner Award, and we had a good time. So watch out for the people you're hearing today. So it really is not just about giving awards. It's great to celebrate excellence, but by celebrating excellence, we also use it to convene leaders. We want to bring the best scientists to Canada to be able to inspire the next generation. And that's what we're here to do today. So what the Gairdner mission stands for is very much in line with promoting the excellence of your research, giving opportunities for us all to learn from each other and take science into the future. So welcome to you all. I'm looking forward to a great day, and I'm looking forward to interacting with the students through posters and through mentoring tomorrow. So I'll hand it back to Jeff.
Okay, so we're ready to go. We have a really action-packed day for you, so one of my duties will be to keep us on time. But that said, we have a lot of time and a lot of places to make up time, so don't feel you have to hold any questions back or any discussion. So it's with pleasure I invite Dr. Tamara Ogilvie, Professor of Biochemistry and Medical Genetics and Kennedy Research Chair in Neuro-Oncology and Stem Cells, to the podium. Tamara.
So good morning, everyone. I'm very pleased to introduce Dr. Mathieu Lupien. He's a senior scientist at Princess Margaret Cancer Centre and also a professor at the University of Toronto. And prior to joining Princess Margaret in 2012, Dr. Lupien earned his PhD in experimental medicine at McGill University and carried out postdoctoral training in medical oncology as an Era of Hope Fellow at the Dana-Farber Cancer Institute, Harvard Medical School, followed by a PLDA, so a program in leadership development at Harvard Business School. I learned a little bit about this program last night at dinner, and it sounds quite fascinating. Dr. Lupien's research in chromatin and epigenetics has pioneered the study of the non-coding genome to identify genetic determinants of oncogenesis and accelerate the development of chromatin and epigenetic-based precision medicine designed for cancer patients. I may be a little biased, but I think the brain tumor work that he has published recently focusing on chromatin accessibility and identification of an invasive glioblastoma stem cell state is particularly fascinating. Dr. Lupien is the recipient of many, many awards. I'll name a few. There's too many to name here, but I'll just name a couple. He has won the Canadian Cancer Society Bernard and Francis Dorval Award for Excellence. He is a two-time recipient of the Till and McCullough Discovery of the Year Award, and not one, not two, but three-time recipient of the Investigator Award from the Ontario Institute for Cancer Research, so very highly accomplished. To give you a quote, I was looking through Dr. Lupien's website, and I found this one to be really very interesting. He says, "We embrace diversity as our greatest asset. It spurs our creativity, galvanizes our collaborations, and drives our innovations. And your lab has certainly led by example." I believe that this is Dr. Lupien's third or fourth talk in third in Manitoba in the last couple of years. So I'd like to say welcome back to Dr. Lupien, this very highly accomplished scientist and probably now an honorary Manitoban. Okay. Welcome.
Well, thank you very, very much for the invitation and for the amazing introduction. I'm almost coming up here with shyness, wanting to hide as opposed to anything else because it's almost too much of a presentation I have to deliver, right? So that's a problem. When you have all these awards, you have to deliver. All right, so that's what I'll try to do today at my best. The talk will be focused on cancer. The talk will be focused on cancer driver. But what I really want you guys to start thinking about is not so much cancer per se, but what's the relationship between phenotype and genotype? To which extent is a phenotype driven by the genotype? And what do we mean by genotype, okay? And so I'll be giving you a talk on cancer, but this relates to any given trait you might be studying, any given disease you might be studying in which there's inheritance involved, right? And so let's get to this with a focus on cancer. So many of you are probably very familiar with this concept that cancer is thought to be a disease that develops from a serial accumulation of mutations. So genetic variants, right? So somatic mutations or inherited variants are fueling the transformation of a normal cell and it to be able to achieve a cancer state. And again, this is going to be achieved by the serial acquisition of genetic variants, the first ones allowing for immortalization, and then the subsequent ones allowing for transformation. And that's more or less the current dogma, which implies something very simple. It implies that for each tumor, for each cancer type, there has to be a single genotype, right? There has to be a unique genotype, because if you end up with a specific phenotype, let's say a lung cancer versus a brain cancer, which are very different in phenotypes, that would underlie that if mutations are driving this phenotype, that they both have a unique genotype, okay? And so there's been tremendous efforts in trying to identify the genotype specific to each cancer type, to each tumor that arises in a patient. And this slide is more or less a summary of recent efforts, international efforts in which Canadians were involved, where the effort was specifically focused on sequencing thousands and thousands of tumors in order to identify the genotype specific to each cancer type. And so this figure more or less has on the x-axis different cancer types. Apologies if it's not really readable, but trust me, each one of these are different cancer types. And the y-axis is the number of mutations one finds in an individual tumor per megabases of DNA. And so in this figure, each small gray dot is an individual tumor. The red line is more or less the average for that given cancer type. So that you can see that there's variability. Some cancer types have low levels of mutations. Some cancer types have high levels of mutations. So to no surprise, on the far right side, it's melanoma, right, caused by UV, DNA damaging agents. So that's normal that there's a huge burden of mutations in that type of cancer. And on the far left, those are typically pediatric types of cancer, right? Pediatric brain tumors, for instance, and the likes, arising early in life, so not enough time to accumulate a large burden of mutations. All right. So the drive for a genotype-driven phenotype has been launched, right? We have all of these mutations. Now, not all of these mutations are going to be directly contributing to the phenotype. Some are going to be passenger events, right? Consequences of oncogenesis as opposed to causes of oncogenesis. And so the next phase of information that needs to be extracted from such data is to discriminate which of these mutations are favoring cancer development versus which ones are just passengers, so that we can fine-tune an identification of what are the genetic drivers, the genotype specific, to a given cancer type, okay? And the way this is accomplished is typically done first and foremost based on a statistical standpoint. So what is done is that one partitions the human genome into partitions of interests. So for instance, one could decide to only look at genes. So there's 26,000 genes or so to look at. So that's a subset of the human genome. So you've partitioned the genome according to genes. And then what you do is when you look across genes, you ask yourself, is there one gene that has a burden of mutation that's far in excess of what one would expect by chance? Is there one gene that has an excess of mutations beyond the null expectation? Okay? And so the examples that are presented here, the schematic representation, so the top one, so this beige rectangle is meant to represent a gene. On the left is the observed number of mutations in a given gene. On the right would be what would be expected by chance for a given cancer type or a given patient. And what you do here is that you simply count the number of mutations per base pairs of DNA in a given gene, and you compare that to what you see on average across all other genes. And for a gene to be listed as a cancer driver gene, i.e. a DNA sequence that contributes to the phenotype, it has to have a burden of mutations in excess compared to the null expectation. So in this case, we have 9 mutations spread across the gene compared to the null expectation being around 1. So this gene, whatever it is, would be classified as a cancer driver gene. Okay? Now there's variant approaches to looking at enrichment of mutations within genes to find additional cancer drivers than those that would be picked up purely based on more mutations over a stretch of DNA. There is this concept of potentially also looking for mutations that are biased towards the same residue. Okay, so if there's a clustering of mutations, you're going to say, aha, there has to be a functional reason for all these mutations to pile up on top of the same residue across individuals, across tumors. And so this would, despite the fact that there's as many mutations on this example as there is on the right side example, this would classify this gene as a cancer driver gene because the mutations are specifically targeting a unique set of residues, so there's clustering of mutations. Okay? And so the 2 examples here, the top one, for those of you that are familiar with cancer, would be typical of a tumor suppressor gene, which is that you can find one of multiple ways to destroy the gene function. And at the bottom, this would be more typical of oncogenes, so genes that promote oncogenesis, and that these mutations would typically pile up on a specific nucleotide in order to change the function of the protein product. So for instance, to create a constitutively active form of the protein. Okay? So this has been around. It's an effort that's been led in recent years to great depth. And once you have, and that has identified hundreds and hundreds of cancer driver genes across different cancer types. And what you want to do when you have these cancer driver genes is that you know that you have a specific DNA sequence that's promoting oncogenesis, that's contributing to oncogenesis. And so the next step in terms of precision medicine is that you want to use that information and match it to drug sensitivities. So that this way, when you scan a tumor for mutations and you can identify a cancer driver gene in a given tumor, you can ask yourself, is there a drug that could work in that patient? Are the presence of these mutations in that given gene, in that cancer driver gene, indicative of response to a given treatment? And that effort is ongoing, and actually there's a resource, an online resource, OncoKB, that captures all that information live so that it's updated on a regular basis. And about a year ago, so this is from February 2021, the number of genes, cancer driver genes that had been assigned predictive sensitivity to a given treatment are listed here. There's 155 of them that were identified. So that's about a year ago. 155 cancer driver genes had been assigned to be a predicted response to a given treatment, actually to one of multiple different treatments, such that we were at a level of close to 567 clinical implications. So that implies a clinical implication is one cancer driver gene being assigned to one drug, sensitivity prediction, okay? And so if you have 567 and you only have 155 genes, that means that same gene is often associated with response to multiple different drugs, okay? So that was about a year ago. And so this is the concept of promoting precision medicine from identifying cancer driver genes out of mutations. Now, if you were to go on that website today, or let's say a couple of months ago in May, okay, that's the last time I took this picture, what you would see are these numbers. So from about a year ago, we had 155 cancer driver genes associated with a predicted response to a given treatment. A year after, we are at 160. So within the last year or so, we've only added 5 genes. Only 5 cancer driver genes have been added to this database as predictive of response to one treatment or another. And I don't know for you, but from my perspective, that's not enough, right? Because there's so much heterogeneity in the tumors across individuals that you want that number to be as high as possible. Like there's 26,000 genes. You don't want to be only using 160 of them in order to predict response to a given treatment in order to be able to guide treatment decision. You want that number to grow. And so the rest of the talk is going to be focused on how can we grow that number? How can we find additional cancer drivers to fuel in this pipeline of matching cancer drivers to predicted response to treatment so as to be able to truly have a huge database to tap into when it's time to figure out what's the best treatment strategy to give to a patient that presents with a tumor. Okay, so how can we do this? So there's 2 concepts that I'll move forward. The first one still relies on genetic variants, on mutations, right, and inherited variants. But tries to expand the search for cancer drivers beyond genes. So why would we want to do that? Well, this figure summarizes the why pretty effectively, in my opinion. The black circle, the big black circle, corresponds to the sum of all bases in the human genome. So you're probably familiar, right? How many bases are in the human genome? 3 billion, correct? And we're diploid, so 6 billion per cell. So this black circle is meant to represent the 3 billion bases of DNA found in any individual cell. The proportion that's coding is showcased here with the purple circle. That's about 1.5% of the DNA in the human genome. It's a small fraction. There's 98.5% of the genome that's non-coding. That's not been mined for cancer drivers, okay? Should we look at the non-coding genome? Is there value in looking at the non-coding genome? Or has the scientific community that has put forward the gene-biased and centric view of the world correct? Okay, I like the response. Perfect. So what you've just heard is exactly it. There's a whole lot of functionality in the non-coding genome, okay? And let's go at it step by step. So the first level of evidence for function outside of genes, outside of coding sequences, comes from simply looking at sequence conservation across species. And so specifically here, the red circle represents the proportion of the human genome that shares high degree of similarity with mammalian species, okay? And as you can see, that circle is bigger than the purple circle. So there's more than just 1.5% of the genome that has conservation across species. There's close to 14%. And as you can see from this as well, most of it is outside genes, outside coding sequences. So just from sequence conservation across species, there's a rationale for looking at the non-coding space. Now, if you're a little bit more permissive, I like to be permissive sometimes, and in this instance, I think it's very much warranted. If you want to be more permissive in assigning potential function to the genome, you can simply start looking for biochemical activity. By biochemical activity, I mean transcription, like RNA production. I mean DNA protein binding, right? Transcription factor, RNA polymerase, DNA polymerase, name it, binding to the DNA. If you start taking into account these types of biochemical activity, from a conservative estimate, so the dark blue circle, you get about 20% of the genome being potentially functional. If you're more lenient in your analysis, you lower your p-value cutoff, you can get a number that's close to, if not even exceeding 80% of the genome. So there's a huge untapped territory in the human genome, beyond genes, that could harbor these cancer drivers, these drivers of phenotype, okay? And so just to give you an indication of what could be potentially functional in a non-coding genome, I'm going to use a gene-centric perspective so that this way we are all anchored on a gene, so it's something familiar. But what we know in the non-coding space that is critical for genes to work are cis-regulatory elements. So for instance, for a gene to be expressed, it needs to have a promoter. That's non-coding, but that's a site where RNA polymerase II will be able to bind. For a promoter to be active, in many instances, it needs to interact with distal cis-regulatory elements, such as enhancers, repressors, anchors of chromatin interactions, and so on and so forth, that are DNA sequences far from the promoter. They might be within the gene, they might be upstream, they might be in introns, they could be anywhere, but they're still non-coding. And these are essential docking sites for proteins, the enhancers being specifically docking sites for transcription factors. And for a gene to be expressed, proteins such as transcription factors need to be recruited at enhancers. They need to get into contact with the promoter that's bound by RNA polymerase, as showcased here at the bottom. Communication happens. There's a signal that's shared. Bingo, RNA polymerase transitions from initiation to full elongation. And you have transcription, okay? Perfect. So could we look for cancer drivers in the non-coding space? Could we apply the same concepts that have been put forward to find cancer drivers within genes to the non-coding genome? And the answer is yes. There's 3 methods that have been put forward in recent years, to which my group has contributed, and others. The first concept is referred to as cancer driver CRE, so a cancer driver cis-regulatory element, that is a single docking site for one transcription factor, for one RNA polymerase, being burdened by mutation more than expected by chance. These are rare, but there's a beautiful example in the literature, which is the TERT gene promoter. The TERT gene encoding for the telomerase, essential for cancer cells to proliferate ad libitum. And so mutations in the promoter of the TERT gene have commonly been found in some specific cancer types to be more than expected by chance. So the promoter of the TERT gene is a cancer driver in a subset of patients. A second approach that's been put forward is called cancer driver regulatory plexuses. So if you remember from the previous slide, I showed you that one promoter interacts with one enhancer to regulate one gene. The reality is that there's more than just one enhancer typically involved. There's 5, 10, 20 enhancers that come together to influence the expression of a given gene by interacting with its promoter. That complexity of interaction is known as a regulatory plexus. And so what you can do is simply question the burden of mutations within the regulatory plexus of your gene of interest versus all other genes in a given tumor. And there's case example in breast cancer and in prostate cancer that you can identify cancer driver regulatory plexuses. That is a burden of mutation in the sum of all regulatory elements of a given gene to promote overexpression of that gene, for instance. Okay? The third aspect is known as cancer driver cystromes. And what is meant here is that there can be a burden of mutation in the sum of all binding sites for a single transcription factor in one tumor or in one cancer type. And so what I mean by this, so I showed you again, for a gene to be expressed, there's a need for a transcription factor to bind to a enhancer in order to communicate with the promoter and induce gene expression. Well, the reality is that a transcription factor is not responsible for the expression of a single gene in a cell. It typically regulates the expression of hundreds of genes in the cell, and it does so by binding to multiple different locations across the genome. And so if you look at the sum of all these binding sites, that's the cystrome of a given transcription factor. Each transcription factor has its own unique cystrome. And so you can simply question what's the burden of mutation in the cystrome of transcription factor 1 versus the burden of mutation in the cystrome of transcription factor 2 to identify cancer driver cystromes. Okay? So from a genetic standpoint, looking at genetic variants, mutations, inherited SNPs, and the likes, there are ways to expand our search for cancer drivers from genes to multiple different functional units in the non-coding genome, okay? But that's still the genetic approach, okay? And so what I want to do now is shock you a little bit more, okay? Could we find cancer drivers without relying on genetic variants, without relying on mutations, without relying on inherited variants, okay? And the reason I'm asking this, you may expect, is because I have a strong propensity to think that the answer is yes. Otherwise, I wouldn't be covering this topic. And I think it's possible because of the fact that the DNA in our cell is not found in the nucleus as a naked form. It's actually found in a complex known as chromatin, where it forms a complex with specific proteins, histone proteins. And typically what happens is that the DNA will be packaged with the histone proteins in a way to form the basic unit of the chromatin known as the nucleosome, which is a sum of 8 histone proteins coming together with 147 base pairs of DNA, okay? So this is showcased here on the far right. So we have 147 base pairs of DNA, the black line, wrapped around an octamer of histone proteins that forms the nucleosome. And the nature of the chromatin state can vary. There's not just one way of packaging the DNA. There's multiple ways of packaging the DNA, which defines different chromatin states. So what I'm showing you here, for instance, at the upper section and the bottom section, is the same stretch of DNA, but in 2 different cells, let's say neurons and muscle cells. And so at the top, what you can see is that that stretch of DNA has multiple nucleosomes. The DNA between nucleosomes is very short. That's known as linker DNA. And when the chromatin is found in this level of compaction, it's said to be compressed, inaccessible for transcription factors for a DNA binding protein, RNA polymerase, name it, okay? In the second cell, that same stretch of DNA can adopt a different chromatin state. And so here what you can see is that the linker DNA between nucleosomes is much, much, much longer. And suddenly, that DNA sequence is easy to read by transcription factors so that you can have the recruitment of transcription factors. So chromatin state 1 prevents interactions with proteins. Chromatin state 2 enables interactions with proteins. So if you think of this, that means that this stretch of DNA is silent in one cell type, let's say neurons, and it's active in another cell type, let's say muscle cells, okay? So these are known as chromatin variants. These types of DNA sequences that adopt a different chromatin state across cell types, cell states, and so on and so forth are known as chromatin variants. And that's the reason that you can, from a single genotype, establish the thousands of different phenotypes that you see in a multicellular organism like we are, right? If I was to sequence the DNA of your brain cells and then sequence the DNA of your muscle cells, I could not distinguish their origin, okay? I could if I sequence your T cells, that's it, practically, okay, because there's rearrangements there. But for the most part, the DNA sequence remains the same. But if I was to look at the chromatin in your neurons versus the chromatin in your muscle cells, I could tell you where that sample is coming from, blinded, okay? Perfect. Here's a little bit more hardcore evidence of this concept, okay? Real data that was published by colleagues. So at the bottom here, we have different cell types, tissue types, color-coded, okay? Yellow, trophoblasts, CD8 cells in orange, and so on and so forth. What we're looking at here is different stretches of DNA across the human genome. And so each shaded gray area is about 200 to 300 base pairs in size. And each line down here at the bottom is showing you this level of chromatin accessibility, so distances between nucleosome, across these different DNA sequences, across all the different cells and tissue type listed here on the far right. And so if I take a look at this first region, for instance, what I would notice is that that DNA sequence is compacted in all tissue cell types looked at except one. I only see a signal in this first line, a yellow signal. So this peak indicates that the chromatin is accessible. And so what this is saying is that that piece of DNA, that 200 to 300 base pair long piece of DNA, is accessible, potentially functional, only in trophoblasts. So this would be a chromatin variant specific to trophoblasts. And in contrast, if I go down here under number 7, the shaded area, again, a 200 to 300 base pair fragment, is listed as being compressed in all cell and tissue types, except one, the orange tissue, which is CD8 T cells, okay? And so then that would classify this DNA sequence as a chromatin variant specific to CD8 T cells. And now, there's not just one sequence of DNA in the genome that's CD8 T cell specific. There's not just one region that's trophoblast specific. Up here, you're looking at all the regions of the genome picked up as accessible in at least one of the different cell types and tissue that were processed in this study. So there was close to 800 different cell and tissue types that were looked at. And you can see here that, so each column is a different genomic coordinate of 200 to 300 base pairs in size. And you can see the first grouping here of yellow sites. These are thousands of coordinates that are preferentially uniquely accessible in trophoblasts. So chromatin variants specific to trophoblasts. The second group in orange, those are coordinates of chromatin variants specific to CD8 T cells. And so this would underline that the DNA sequences there are likely drivers of trophoblast phenotype or of CD8 T cell phenotype, okay? So that's from normal biology. Could we consider cancer arising free of genetic variants and dependent on chromatin variants? That would be a major game changer, wouldn't it, right? Because there's been this strong genetic bias that one genotype, one phenotype in the cancer field. So could we show that cancer can arise free of genetic variants that are driving the phenotype, but instead relying on chromatin variants that are driving the phenotype? Well, this is something that there is one example in the world that it may be happening. And I say may because as a scientist, we're always not in agreement with the working model until it's proven and proven again, right? And so here's an example. So this was work led by our collaborator at Sick Kids, Michael Taylor, who had an interest in studying the genetic basis, the drivers of oncogenesis in a specific form of brain tumors in kids known as pediatric ependymoma. And what Michael's group ended up doing was whole genome sequencing, whole exome sequencing to find the genotype driving that phenotype, driving that type of brain tumor. And what they ended up discovering is that there's no genetic drivers, there's no drivers of oncogenesis that they could identify from mutations, okay? And this is depicted on the left figure. So the red line is showing the mutations identified per megabases. Ependymomas are down here at the bottom, and this is in comparison to other cancer types, glioblastoma, brain cancer, and so on and so forth. Melanoma, right, the number one disease with the highest burden of mutations. And what you can see from this line is that pediatric ependymoma don't really have that many mutations per megabases. They're on the low end, very, very, very few mutations. The blue histograms, what they're doing is that they're reporting cancer drivers, cancer driver genes specifically. And so what was done here was taking into account the mutations, assessing whether they would be burdening one gene as opposed to another. And as you can see, glioblastoma has a large number of cancer driver genes, so many mutations targeting specific genes, and so on and so forth. But down at the ependymoma level, nothing. Not a single cancer driver gene could be identified. And so that led the question as to whether that type of cancer could be driven by chromatin variants as opposed to genetic variants. And so one of the different ways the chromatin was studied, and one of the different ways chromatin states were measured in order to identify chromatin variants, was to look at DNA methylation. And what we're seeing here is levels of DNA methylation across thousands of different genomic coordinates, in yellow implying that there's a high level of DNA methylation, in blue implying that there's a low level of DNA methylation. And if you compare DNA methylation level, you're more or less looking at two different chromatin states, highly methylated or unmethylated, looking for chromatin variants. And in doing so across a pool of patients, so each column is a different patient, they ended up realizing that you could stratify this disease into two different groups. So there were chromatin variants distinguishing two different types of pediatric ependymomas, labeled as group A CIMP positive and group B CIMP negative. And so this one, I'll just pay your attention to this, has a high level of DNA methylation. Most of the chromatin variants showcase an increased abundance of DNA methylation. And to showcase that this was functionally relevant, Michael's group came back in and simply treated pediatric ependymomas with chemical inhibitors of DNA methyltransferases. So to eliminate DNA methylation, to reverse the chromatin variants back to an unmethylated form. And in doing so, looking at cell survival, looking at ability to engraft and form tumors, what they could see is that the group A tumors, those that had a high DNA methylation associated chromatin variants, were more sensitive, highly sensitive to DNA methyltransferase inhibitors. So that's showcased here by the black and the orange line in comparison to the purple and the green line. And same thing here in the limiting dilution assay, the orange line being much different than the control lines, the black and the gray line, in their ability to engraft and form tumors after being treated with DNA methyltransferase inhibitors. Okay, so functionally validating the role of chromatin variants in enabling the oncogenic phenotype of pediatric tumors. Okay, good. Now, some novelty. Could we use chromatin variants to go beyond what was just described and truly find genetic drivers of oncogenesis? And here I want to draw your attention to specifically to stemness properties, okay, in cancer. And for stemness property, one of the best models to study is hematopoiesis and blood-related cancers. Those of you unfamiliar with hematopoiesis, this is the lineage tree commitment that has been established in the past, which has at the apex stem cells. So this is for normal blood production. So at the top of it all, it's long-term hematopoietic stem cells, followed by short-term hematopoietic stem cells, and then a collection of progenitor populations. So they're not fully differentiated. They're not fully mature cells. They're on their way to becoming mature. They're still somewhat able to give rise to one or multiple different mature populations. And then at the bottom, you have all the mature cells, the B cells, the T cells, the natural killer cells, and so on and so forth. Okay? And so the key take-home message from this slide is that you have a collection of populations that have self-renewing, stem-like property, progenitor-like property, versus mature populations. Now, we were fortunate enough to work in collaboration with John Dick on this type of populations to characterize the chromatin state across the genome of each of these different cells and hunt for chromatin variants. Okay? And here's one of the interesting observations we made. We picked up chromatin variants that specifically distinguished the mature populations from the progenitor and the stem population. This was a black and white type of observation, very shocking to me. And so, again, here I'm showing you the differentiation tree in normal hematopoiesis, color-coded for stem, progenitor, and mature population. And if you look on the right side, I'm showing you 4 different locations on the genome, roughly 200 to 300 base pairs in size, where the signal presented here is reflective of chromatin accessibility. And so the higher the peak, the more accessible the DNA is. If there are no peaks, it means that the chromatin is compacted, that DNA is inaccessible. And this is black and white, right? This is, I mean, I look at it today again, and I'm mesmerized by the cleanliness of these results, right? You can see peaks in all stems and all progenitors, but 0 peaks in the mature population. So these are primitive cell-specific chromatin variants, okay? And so these are 4 of these. What's intriguing about these regions is if you look at the DNA sequence they harbor, you have to ask yourself, what's the DNA sequence there, right? What turns out to be present in these 4 regions are listed here, LTR33, HERV3, LTR16, E1, HERV3, and so on and so forth. You might never have heard of these, but if you have, this should raise huge interest. These are repetitive DNA sequences, so parts of our genome that are highly repetitive, okay? This is the genome, right? I told you about coding. 1.5% of the genome is coding. I've told you that 98.5 is non-coding. What scientists typically study is barely 45% of the genome, which is non-repetitive, okay? What we typically exclude from our research are repetitive DNA sequences. Those account for 55% of the human genome. You might have heard this year about the T2T project, the telomere to telomere genome project that finally released a complete genome sequence. The challenge that we had before releasing is these repetitive sequences. They're super hard to sequence. And so that's why most of the research that people have spent in assessing the function of the non-coding genome has been focusing on the non-repetitive space. But suddenly, from the data that I just showed you, and from others in the space, there's interest at looking at the non-repetitive, at looking at the repetitive genome. And so the repetitive genome accounts for about 55% of the genome, and I've labeled with the red stars the sections of the repetitive genome that actually harbor a specific type of repetitive sequences that we're interested in, which are known as transposable elements, okay? And so those account for roughly 45% of the genome. And so those are the ones that we see in chromatin variants that distinguish the primitive from the mature populations. And these have been studied in the past in developmental biology for the most part. So there's a huge space of research, Barbara McClintock having been one of the pioneers in that space, that has characterized the role of transposable elements in biology. They've been picked up as transcribed DNA sequences with function. They've been picked up as active cis-regulatory elements. And in more recent years, the space of function has also been growing in the cancer arm of research, where we've seen them, again, have a role in oncogenesis while being transcribed or while serving as cis-regulatory elements, okay? So there's a huge space of interest. Now, here's the deal with regards to stemness. From the observation that I showed, we went deep and decided to fully characterize what are the transposable elements specifically accessible uniquely in the chromatin variants of primitive versus mature populations, okay? Now, this is a complex figure. The key take-home is right here on the right side. Look at the order of families listed here, okay? We've ranked them according to transposable element superfamilies that are preferentially accessible, preferentially found in chromatin variants specific to primitive populations versus chromatin variants specific to mature populations. So at the top, we have the HERV1, HERV2, HERV3 line elements, and at the bottom, we have the sign elements, sign 1, sign 2, and so on and so forth. Okay, so remember that, okay? Put that in your mind for a few seconds. HERV1 line, like HERV and line elements first, sign at the bottom. This is for normal hematopoiesis, okay? Now let's look at cancer, leukemia, AML. You can partition from a patient sample leukemia cells into multiple different fractions based on cell surface markers. The field has identified CD34 and CD38 as a mean to enrich for leukemia stem cells as opposed to non-stem cells part of the AML populations, okay? And from these fractions, in partnership with our collaborator John Dick, two things can be done. A fraction can be used to profile chromatin states and map chromatin variants. And another section of the cells can be used in limiting dilution assays to functionally validate the leukemia stem cell phenotype of a given fraction, okay? And then we can make comparison. So we've done that over close to 100 samples from close to 25 different patients. Leukemia stem cell positive fractions are in red. Leukemia stem cell negative fractions are in dark blue. And we here show the stratification of these fractions according to the chromatin variants found within each of these samples over repetitive DNA sequences. And just look at the dendrogram. It's black and white. The first separation is perfectly taking apart LSC positive versus LSC negative. All the LSC positive are on the left side. None are on the right side. Black and white. It's unbelievable. Like I was shocked when I saw that, and I'm still shocked. But here, with regards to what I just showed you a second ago, look at this order. Again, we've ranked all these transposable elements superfamily according to them being preferentially found in chromatin variants specific to leukemia stem cell positive fractions versus leukemia stem cell negative fractions. It's the same order as what we saw in normal hematopoiesis, right? The HERV elements are at the top, line elements, followed at the bottom with the sign elements. same pattern that we see between normal development as we see in leukemia. Wow, okay, what do we do with this, right? Because what we're doing there is simply using chromatin variants and assessing statistical enrichment of chromatin variants over specific DNA sequences. And so we're identifying DNA sequences of interest as cancer drivers. And so in this case, we're finding transposable elements from the HERV family, from the line family, as putative cancer drivers of a stem phenotype. So how can we actually transition from just listing them as putative cancer drivers and showing that they actually are functional drivers, okay? And this is where genome engineering comes into play. You are likely very familiar with genome editing, which uses the CRISPR-Cas9 technology to go and cut and change the sequence at regions of interest. Well, what I like to do is genome hacking, okay? Which does not involve changing the DNA sequence, but rather changing the chromatin state. And the way this can be achieved is that we still use the CRISPR technology, but with a dead Cas9 part, so that there's no more an ability to cut the DNA. And instead, the Cas9, the dead Cas9 is fused to a chromatin factor, an enzyme that can change the chromatin. So a DNA methyltransferase, for instance, a histone acetylase, a histone deacetylase, name it, whatever you want, okay? And so this allows you to maintain the DNA sequence intact, but change the chromatin state, and therefore determine whether the chromatin state is a driver of phenotype, and if the sequence, the endogenous sequence within the region you're targeting, is contributing to the phenotype. Now this is the second to last slide. I'm going to show you some super preliminary result to take with a grain of salt, but still the concept is, let's do this in leukemia stem cell positive fractions. Let's change the chromatin states at regions of interest, at regions that we identify as cancer drivers, and in such a way to transition, let's say, a region that's accessible into a region that's compacted, and let's assess impact on phenotype, okay? And so this, again, is preliminary result, and the data came out from the lab yesterday, or 2 days ago. So again, to take with a grain of salt, but super promising. What we've done is that we've worked with a model cell line of leukemia, which is far from an ideal cell line. It's not the leukemia stem cell positive pure fraction, but it's better than nothing, so it's a first step. So the MAL13 cells. And what we've done is that we've actually used genome hacking approaches to target transposable elements, specifically accessible in leukemia stem cells fractions, okay? So we're hacking what we think are drivers of oncogenesis, these transposable elements. And so we're doing so in a way that enables that region to transition from being accessible to becoming fully compressed, losing its ability to recruit transcription factor, be transcribed, and so on and so forth. And the results shown here are pretty striking. After a couple of days, 3 days, 4 days, and up to 7 days of assessing growth in cells in which we've targeted one specific transposable element family, the LTR12C, we can see a significant drop in the number of cells. And so now we're moving forward and applying the same concept, but with models that are actually enriched for leukemia stem cell positive cells in order to really be able to assess the impact of changing the chromatin state over unique transposable elements in the optimal conditions possible, with the hope that the impact will be even more drastic than what we're seeing here. We're also hoping at one point to be able to target more than just one transposable element, because there is more than just one found within the chromatin variant specific to the leukemia stem cell fraction. Okay, good. So take home from this talk, if you're to try to understand the contribution of the genome towards any phenotype of interest, please don't just look at genes to find the genetic drivers of the phenotype, okay? Look in the non-coding space. If you're to look for drivers of phenotype, keep in mind that any individual phenotype is a phenotype of genomic variation. It is not a phenotype purely of genetic variants, but it can also rely on chromatin variants, okay? So don't just look at mutations, also take into account transitions in chromatin states. Another point of interest, the repetitive DNA sequences, transposable elements, for instance, can be drivers of phenotype, okay? We shouldn't dismiss them. There's opportunities to start looking at them based on technology having improved significantly to map and sequence over repetitive sequences. Please explore that space. And finally, genome engineering. Your generation is going to be transforming research through genome engineering methodologies. There's not just genome editing. There is also an ability to change the function of DNA sequences by affecting their chromatin states, so don't shy away from using those methodologies. All right, so with this, I'll thank the collaborators, folks from my group that did the work, so Giacomo Grillo, fabulous postdoc in the lab, Bettina Nadorp, that have spearheaded the work that I showed you on leukemia and hematopoiesis. This was done in collaboration with John Dick, Helena Budzen from her group being a driving force, Jean Wang and Mark Minden, and of course the funders, CIHR, OICR, CERC, Stand Up to Cancer, TFRI. Have been very fortunate to have support from many different institutions, so a huge thank you to them, and a huge thank you to you for paying attention to this talk. Thank you.
Questions for
Oh, questions, okay.
Come down to the mic, please. Thanks.
Hello, great talk. Thank you very much. I was wondering, for the last slide you presented, wouldn't it make more sense to get a non-cancerous cell line and using the genome hacking to open the chromatin and see if it transforms and makes?
You can do
Cancer
both.
cell.
You can do both, so you shouldn't shy away from doing both. So because in the leukemia system, we can work with purified leukemia stem cell fractions and leukemia stem cell negative fractions, we have the ability to do both. Our thinking is that and we might be wrong, right? This is the working model. We're thinking that it's going to be easier to destroy a phenotype than to create a phenotype, considering the limited ability we have at changing the genome, right? We can still target one TE family at a time, and there's a bunch that need to be accessible, presumably. So we are hoping to be able to see a sufficient drop in the phenotype by eliminating just one, and we are expecting it to be much more challenging to create a phenotype from changing just one. So that's why we've tackled the LSC positive fraction to start with, but you are 100% correct. The genome hacking technology allows you to do both. You can put enzymes as fusion proteins at the CRISPR-Cas9 that are going to be enabling chromatin accessibility or restricting chromatin. You can do whatever you want. It's beautiful. That's the beauty of it.
Great, thank you very much.
Yeah, beautiful. Thank you.
Since we're coming from across Canada, it'd be nice if you could introduce yourself.
Sure. So my name's Ellen. I'm from Queen's University, which is in Kingston, Ontario. Thanks for your talk. This isn't my area of specialty at all, so I'm going to ask you a very foundational question.
Yes.
Are all mutations considered cancerous?
Nope.
So are there ways of distinguishing between cancerous and non-cancerous mutations?
Right. So it's very challenging to do so when assessing the function of a single mutation. You could use genome editing and introduce that mutation, for instance, to assess if it changes the phenotype of your model of choice. That's laborsome, that's very complex, but that's one way. You can potentially infer function from computational methods. So for instance, if you think that your mutation falls in an enhancer, you can assess whether it's found within a DNA sequence that's known to be recognized by a transcription factor. And if it is, then you can compute a likely differential, a likely affinity change introduced by that mutation over that DNA sequence. So you can start inferring potential function, but it's extraordinarily challenging. The approach that has been used by the community so far has mainly relied on partitioning the genome in functional units of interest and not necessarily wondering about the function of an individual mutation, but rather using mutations as beacons of regions that accumulate mutation, and this accumulation of mutations is interpreted as a reason for positive selection.
Okay.
So that's been the easiest approach employed to date.
Okay, thanks.
But yeah, there's a ton of passenger mutations, a ton. And the worst of it, think of this, okay? I'll push that even further. Sorry, I'm not letting you go.
That's okay.
I need someone else on that, okay? So think of
okay.
this, right? For cancer initiation, your cells are going to change their chromatin states over time, right? Because the cell is normal and becomes and acquires a different phenotype. So a given DNA sequence might be accessible in the normal setting, might no longer be accessible once the cell is fully transformed, and vice versa. Now imagine that there's a mutation that's causal. driving oncogenesis, popping up into the normal tissue and driving that first phase of transformation, and then you measure the mutations later on in life. And if you were to look at chromatin states at that time, that same mutation might no longer be in accessible chromatin. It might now be in compressed chromatin. And so it would have no more function. So it might have been historically important, but has lost function. And so that there's no need to keep that mutation around anymore. And so it's a huge challenge to be able to distinguish just from sequencing mutations which ones are drivers versus not drivers, because they might have been driver in the past, but might no longer be drivers now. Same thing happens for what we call passenger mutations. They might be passenger mutations in the context that they populate compacted chromatin at the time point that they're acquired, but then as a tumor evolves from primary to recurrence, or from primary to metastasis, chromatin state's going to change. And so a mutation can suddenly find itself in accessible chromatin and might have a huge impact on the oncogenic phenotype. So this is like a major challenge to address in the future if one really wants to specifically look at individual mutations to account for a cancer phenotype.
Mm-hmm. Okay, thanks.
Yep.
My name is Lily, University of Calgary. Thank you, it was an amazing talk. I was just wondering, for a specific cancer type, how would you decide the location of a chromatin for the genome hacking?
Yeah, so my hope is when I get diagnosed with cancer, right, that's more or less what you're asking, what do I want when I'm going to be a patient, right? And my hope is that my tumor will be profiled at the genetic level to find mutations as much as at the chromatin level to find chromatin variants. And with these chromatin variants, because we're growing the pool of maps of chromatin states across different cancer types, I'll be able to compare it to whatever has been reported in the past in other cancer types, in other normal tissues, and so on and so forth. And maybe if I have a good clinician, I might end up even being able to compare it to the flanking normal tissue of my tumor in order to be able to find chromatin variants specific to my tumor. And those will be regions that I will be able to prioritize for genome hacking based approaches. And you can rank them, like not all chromatin variants are equal. Some are uniformly accessible in your cancer compared to normal tissue. Some are going to be partially accessible, right? Because you're working with populations of cells. So some are going to have, let's say, openness in 50% of your cancer cells, and other are going to be open, accessible in 100% of your cancer cells compared to normal. So I would rank those at 100% first as primary targets. So will that happen one day? Well, we have a lot of lobbying to do. Let's phrase it that way.
Thank you.
Yeah, thank you for the question.
Part of chromosome hacking would be transient, right?
And yes, okay? So I'm all in for that type of CRISPR technology. Like I would take it. Yes.
Thanks.
Thank you.
voting discussion, we're going to break out a coffee break right now. Upstairs in the concourse, we'll start at 10:30 sharp.
Thank you.
So good. You explained everything so training there. That was fantastic.
Thank you.
Yeah.
Good to hear. Good. I spent a lot of time last night reworking the slides.
Oh, it was beautiful. No, you really explained the concepts very simply.
Good. Thank you.
Yeah, it was fantastic.
So now you're all going to do chromatin research, right?
You got me thinking more about it.